Good afternoon, everyone. We've been quite lucky that my improvised technical skills haven't betrayed us this wonderful afternoon of the last day of January 2025. You're never going to have this chance again on this day. So, I know that some of the things I'm going to say could be or may have been seen or heard, but I'm just going to say it again because hopefully it will help reinforce. So let me introduce myself. I'm Dr. Manuel Corpas. I am senior lecturer in genomics, and I've been working on this field for about 20 years. I've got a PhD in Bioinformatics from the University of Manchester, and I have a 4 years postdoctoral experience at the University of Cambridge. And I've been at the University of Westminster for about 3.5 years. For those of you who are interested in big data and genomics, come and have a word with me. I'm happy to discuss options for you, projects, and even careers. So I'm going to make this presentation not very sort of computer data intensive. I'm going to focus more on the sort of biology aspect of the genetic data or genomics data of patients. So I've been working with patient data since 2008, analyzing using next generation sequencing information and data for diagnosis of patients with rare genomic disorders. I was involved in one of the biggest projects in the world to sequence 13,000 children with rare genomic disorders and their parents, and the informatics infrastructure that I developed is still being used as of today. So I'm going to give you some of the knowledge that I have gathered, not just in academia but also in industry, because I also started a company that helps clinics use genetic data to understand the diseases and the genetic predispositions of patients to develop diseases. So I'm going to give you a precision medicine approach to using genomic data. So I will be giving you some guidelines that are used whenever we want to classify a variant to see whether it's pathogenic or not. I'm going to also talk about monogenic variants, but also polygenic variants. I understand you guys have been shown a little bit about monogenic variant, so we're going to see that from my perspective, okay? Which I'm sure will be slightly different from what Dr. Volpi provided. So the starting point is that, and we see that again and again, a lot of the drugs, a lot of the treatments that have been developed to date, they work very well for those populations where those treatments, those diagnoses were developed for, which are usually Northern European white men. And that has tremendous implications in our ability to precisely treat people, because we know that depending on your genetic environment, on your genetic makeup and the environment, there are going to be tremendous influences that will modify the way you are going to react to drugs, the way you are going to get cured, or even the way you are going to express the disease. And thankfully, we are beginning to at least appreciate, there's a lot of work to do, but at least we are able to appreciate that there has to be a much more targeted approach whenever we develop any kind of treatment, targeted to the genetics. And so this presentation, this work I'm going to show you, will give you a state-of-the-art understanding of where we are at with personalized medicine, which for me, for the purposes of this presentation, personalized medicine and precision medicine are going to be the same thing. And it's basically about making tailored and specific interventions, taking into account the genetic and hopefully the sort of familial as well as environmental circumstances of each patient. So I stress again, while we are still far from achieving this goal, at least we have a great goal to target, so that everyone, everywhere, regardless of the socioeconomic status, country of origin, where they live, should have equitable access to healthcare. And that's what I've dedicated my professional life to, in improving equitable access to health. And that equitable access to health is going to depend tremendously on understanding the data that we are able to generate from the patient, because the data is really what defines how we are going to treat these patients, how we are going to identify whether patients are stratified in different clusters depending on their genetic makeup. So there are also quite a few lofty goals here, such as making medicine more participatory, preventative, personalized, and predictive. And in the area of AI, where we are at, there are tremendous opportunities for accelerating these relationships between what we call genotype. Have you heard that word, genotype? And phenotype. So what we see at the level of the DNA, what we see at the level of the actual measurements. And that's really what genomics is about. So genomics and genetics in medicine is about understanding the genetic basis of what's causing a disease, really. What's causing what you see, the phenotype, genotype-phenotype relationships. So we want to be able to provide the right drug to the right patients and at the right time in the right dose. And as I say, this is going to be tremendously influenced by your genetics, because depending on the type of variants, have you heard that word, variants? Depending on the type of variants that you have, your ability to metabolize drugs is going to be affected. There are some genes, we are going to see this later on, but there are genes whose variations affect your intake of drugs, and that could potentially, in some cases, things that work for some ancestries. By the way, I'm not going to talk about race. I'm not going to talk about ethnicity. I'm only going to talk about ancestry, because neither of the other terms, which I'm not going to mention again in this lecture, and all of those have a tremendous amount of baggage, and they are socially and historically constrained and abused. I'm just talking about genetic distance. And we know that geography correlates, so geographical distance correlates with genetic distance. Okay? So people from Africa, let's say, they're going to, or of African ancestry, they're going to have a greater genetic distance from, let's say, British people than let's say English ancestry people, because there are many. Even myself, I get confused with these things. But anyway, but hopefully you get the point. What I'm trying to say is that if most of the treatments, most of the research has been developed in English heritage people from Northern Europe, then the further you are in terms of your ancestry, genetically speaking, to the training data set, the less likely or the less precise are likely whatever diagnostics, whatever treatments we have are going to work. Okay, did I make myself understood? Hopefully that's very intuitive. And as I say, I and many people are working on this to make sure not just that we raise awareness, but also that we bridge that gap, okay? And I gave a presentation probably at the biggest conference on this field in genomics yesterday here in London, and it's not a question of precisely about this topic of diversity. It's not a question of no, there's no will. I mean, there's a tremendous amount of will, but for this type of technology to become accessible to everyone, there are so many hurdles. You know, it's a technology which is, you might agree with me, right? It's a technology which is complex, right? You need to have an infrastructure to run a genetic test. You need to have the appropriate supply chain. You know, it's costly, right? And then it has to be accessible. So there's no wonder that's people in the Amazon are not going to have very good precision medicine approaches, since it's very difficult for that technology to reach there. And one of my greatest message I had to say there yesterday was that, rather than making precision medicine accessible, we need to consider also bringing it to people who are not even able to move from where they are. So that's part of my current predicament. Anyway, back to what I wanted to say. I'm sure that you probably agree with me that, having a genome, having the map of a genome changes everything, or at least it's changed it for me, right? So if you take Google Maps, everyone takes it for granted, but and it's because it's free and we don't have to pay for it. But if they took away Google Maps, I would pay to have it. It's such a useful thing, because it lets you go from point A to point B, and you don't get lost, right? So having a physical map with coordinates allows you to find things, right? So that's precisely what the Human Genome Map or project allows you to do. It allows you to find genes at specific coordinates, at specific chromosomes, at specific locations. It allows you to find regulatory regions. It allows you to find the precise physical locations of mutations. And therefore, because we can identify the precise locations of specific mutations, then we can compare people by looking at that precise position and then understanding the array of different types of potential variations that can happen. We call them alleles, right? You've heard of alleles, right? So we have alleles. We have, let's say that we have, my favorite gene is called melanocortin-1 receptor, right? So melanocortin-1 receptor has one specific SNP. You know what a SNP is, right? Anyone? I really need all of you to know what a SNP is. If you want me to, if you don't know, please, I will explain it. Is there anyone who doesn't know what a SNP is? Okay, so I need to explain it. Very simple. So you know that a sequence of DNA, right? A, T, C, G, E. Sorry, no E. A, T, C, G. A, T, C, A, T, 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 C, G, E. That's the in its raw wildest expression. That's what the genetic code is, right? It's a sequence of A, T, C's, and G's, right? Adenine, thymine, cytosine, guanine, right? So the combination of that set of letters is what gives the genetic code, right? You follow that, right? We all agree on it? Yeah? I want everyone who... You agree on that? Cool. So if at position 1,275,495 in chromosome 1, instead of having a T, we change it into a C, so we call it a point mutation. So one letter at that position, T changes into a C. That's what we call a SNP, a single nucleotide polymorphism. So you can have different forms, shapes, at a particular position. And SNPs is I knew that if I just even went into, I knew I would repeat things, but I know that even if just explain it from my perspective, it's going to be different anyway. I'm not sure if you can see. Can you see a very, very sort of transparent there? But you see T, C, G, A. Can you see that from the back? Not sure. So that's what it looks in a digital format, right? Sorry, I wasn't meant to show it like that, but that's the background I chose. Basically, that's a sequence of DNA, and that's the genetic code. That's what we read. That's what when we genotype. A genotype is the specific combination of sequence that we find for a particular sample, a particular individual, right? So that's the genotype. So a SNP, single nucleotide polymorphism, a single nucleotide polymorphism is the workhorse of genetics and genetic analysis. Right? So if you are doing a PCR, you are likely to try to find a particular SNP. If you run an array, have you heard of arrays? That's one of the most likely type of mutations you're going to work with. If you look at polygenic traits or diseases, I mean, can you all agree that height is a highly polygenic trait? Why? Because you have thousands of genes involved in determining height. But also happens with all of the different types of diseases. I mean, type 2 diabetes is highly polygenic. Atrial fibrillation, everything that most people die in industrialized nations are going to be polygenic diseases, right? Type 2 diabetes, atrial fibrillation, inflammatory bowel disease, coronary artery disease, breast cancer, and prostate cancer, 25% of all deaths are going to have at least one of those. Yeah? Could you agree on it? I mean, you all know someone who will have some of that, right? So, and as it happens, we are able, for the first time ever in history, to really understand what little changes, what little SNPs are actually influencing whether you are more likely to develop type 2 diabetes than not, right? Or even closer to us, right? You remember COVID, right? We all forgotten about it. Let's remember how it was, right? Especially at the beginning. You had people in the same family, same food, same environment, same everything, why were there some people who got really sick and some others didn't? Right? When you have the same environment, there was a genetic component that explained, at least partly, that. Okay? So for the first time, we're able to really identify what is it within the sequence of DNA that is actually contributing to that susceptibility. And that's the great new brave world we live today, in which we are able, for the first time, to quantify the genetic contributions to diseases. And we do that by looking at the comparison that we get at the level of allele frequencies. Yeah, allele frequencies. So for that hypothetical SNP that I mentioned earlier, in chromosome 1, position 1 million, whatever, you have two alleles, right? You have the wild allele, let's say is a T, so that's the wild allele, and then you have the in Mendel's vocabulary, or in sort of genetics vocabulary, you will have the mutated allele, let's say, right? That would be a C. So you have two alleles, and by understanding the frequencies of those alleles, we can then associate whether there's a significant association between the presence of one specific allele and a particular phenotype or disorder. Any questions? I know I've spoken quite technically and quick, so I'm going to give it a little pause. Have I lost some of you? I'm happy to reword, rephrase, rethink. So having a genetic map of the genome allows us to be able to understand how different individuals vary depending on the type of alleles they have. Okay, so as a geneticist or genomicist, you are concerned in understanding the frequency of specific alleles and whether those specific alleles are likely to be associated to a phenotype or not. That's what all comes down to in genome medicine. I don't know what Dr. Volpil told you, but that's how I see it. And I don't think it's probably aligned with what she might have said. So hopefully I've convinced you that there's potential in this type of science. But I haven't really illustrated the fact that this science, this biological science, is so data intensive. It's so data intensive because every trillion of your cells have 3.3 billion letters of DNA, which, I mean, nowadays in the time of AI and large language models, 3.3 billion doesn't sound like that much. I mean, I was working with a large language model this morning of 200 billion tokens. But anyway, 3.3 billion letters in every single nucleus of the cells that have your organism have that. Okay? And so it's impossible for a human brain to really grasp that. That's why, but we are lucky because computers are super good at analyzing strings of letters, which is really what it comes down to, strings of letters. So here is our human genome. We have 23 pairs of chromosomes, and I need to check. Do you know what haploid versus diploid means? Can someone remind me, please? I've got a very short-term memory. What's the difference? Haploid versus...
Haploid is when a cell contains half the DNA content. So in a diploid cell, there will be 46 chromosomes, but in a haploid cell, there will be only 23.
Correct. Yes, so basically we have... our genome is diploid, meaning we have two homologous copies of the same chromosome. Mind you, I didn't say identical, I said homologous, right? There's a slight nuance there. So they are not identical, are they? And actually, one set of copies of chromosomes come from your biological mom, and then the other one comes from dad, right? Your biological dad. So, hence, we might have different alleles in the same SNP, right? Because your parents are not going to be identical in their DNA. And so, that means we're going to have positions that are with different DNA sequence depending on which chromosome you're looking at. So let's say that we have one SNP in position 1 million, chromosome 1. You have, at that position, you have on one chromosome, on one homologous copy, you have a T. In another, in the same position, in the other chromosome, you have a C. So can someone tell me how we would call? So in other words, I'm going to give you a clue. We have something called zygosity, which has to do with whether the allele copies are the same or are different. Can someone remind me of what we call if we have the same allele at the two positions in the same chromosome? What's the word for that? Homozygous. Homozygous. And if we have different? Heterozygous. So that's very, very important, okay? I want you, hopefully you knew that. I'm reminding you again in a different context. Hopefully that's useful. There's also another thing I want to say about genes. Any questions? Let me know if you have any questions before I move on. Any questions? Not yet? Okay. So a gene, right? We have within genes, so I'm going to define for the purposes of this lecture, a gene is a segment of DNA that has a start position and an end position, right? It's just a segment. But we know that there's an internal structure within those genes, which is quite sort of unique of nucleated cells, eukaryote cells. You've heard of that thing, right? That term, eukaryotic cells. So eukaryotic cells, they have more complex structure within their genes because prokaryotic cells, all of the gene gets transcribed and translated into a protein. But that's not the case in eukaryotes, right? And as it happens, human beings are eukaryotes, aren't we? So there are some bits that are transcribed within the gene, and there are some bits that are not transcribed within the genes. Okay, so Have you come across this distinction? Okay, cool. So what do we call the transcribed bits of the gene? No. Exon. And the non-transcribed bit? Intron. So last time I looked at the genome, if we put together all of the exons that there are in the about 20,000 genes that we have, so there are around 200,000 exons. So in other words, on average, although the variability and the sort of spread is enormous, but because there are genes that have only one exon, there are genes that have 20 exons. But on average, there are around 10 exons. I wouldn't say average, the median is 10 exons per gene. Right? I think the average might be like 7. But, we have, most genes have many exons, okay? And, can someone tell me what the effect of exons have in terms of the products of a gene? So what happens within the cell that by having different exons allows you to have more of?
Codes for amino acids, proteins.
No, I'm going into, have you heard of something called alternative splicing? Cool. Can someone remind me what alternative splicing is?
Sometimes some exons or introns might be skipped over, so then you end up with different isoforms of product.
Exactly. We have, so we can call them as well transcripts, right? So that level of complexity of having introns and exons allows you to have X number of isoforms, you call them, X number of proteins in the end, depending on the type of exons. And so I'm actually assuming, I haven't said it, but I'm assuming we all know that what the central dogma says, right? Central dogma of molecular biology, which is not so dogmatic because there are many different sort of variants, but I think, it works kind of. So gene encodes for RNA, RNA encodes for protein. So the sequence, its most basic level, the sequence of DNA is going to determine the sequence of amino acids of the translated protein, and that sequence of amino acids will condition the structure of the protein, and the structure of the protein will condition the function of the protein, right? I mean, this is kind of a simplistic way of explaining it, but, it's good enough as a working definition right now, okay? Because, biology never has absolutes. There are always sort of exceptions to the rules, but this is a good model to follow. We have a sequence that conditions the protein structure, and the protein structure conditions the function. So if we have a catalytic site whose coding whose active site is an amino acid, and that amino acid has been changed, phenylalanine has been changed into a tryptophan. So that will have tremendous implications in the structure of the protein. It might truncate it. It's certainly going to change dramatically how that protein folds, and as a consequence, you're going to have different function, if any, for that catalytic site. So you can see that all these different changes that finally, or in its last sort of result, come from the sequence of DNA, then that's going to condition down the line the function of the cell, and as a consequence, the tissue, the organ, and the organism, potentially. Okay? So, hopefully, we now know that genes have exons and introns, and that the different combination of exons and introns are going to have different protein shapes, different protein structures. And that allows us to have, instead of just 20,000 potential proteins, you end up having 200,000 different types of proteins. One gene can have, if you look at ensemble, which is the sort of map of annotations of the genome that I use, you can have a gene that has 10 transcripts, and each transcript is going to have a different function potentially because it has a different combination of exons. All right? Any questions? I'm going too slow, I know. So the Human Genome Project, basically the idea is to have, we all have a different genome, okay? My genome is different from your genome, and yet we are 99.5% identical. So there's a lot of variation in just 0.5% of the differences. And the differences in our genome, those small differences is what makes me be a male, or have dark hair, white skin, whatever, green eyes. So, in reality, what we use as a human genome is just simply a consensus across the whole length of all the chromosomes, but by no means is representative of humanity itself. Because, the best possible reference genome that we could have is your own. But actually, in order to make things slightly easier, we end up having some kind of consensus sequence that then we compare against. So if for a particular position my genome is different than the reference sequence, I call that a variant. Okay, so this concept of variant is very important in genomics. All right? A variant is a difference that a genome has with respect to the reference genome at a particular position. So given the same position, thankfully now we have coordinates, so we have specific positions whereby we can compare the genomes of individuals. And whatever differences we observe between the genome of an individual with the reference genome, that's what we're going to call a variant. And an example of a variant is a SNP, right? And on average, a human genome has around 6 million SNPs. Okay? 6 million SNPs. So you try to analyze every single SNP by hand. There are thousands of something called insertions, deletions, and something called copy number variants. Have you heard of those things? And then something called structural variants. Hopefully you've heard of those. So there's something called ensemble.org where you can actually browse the genome. You can zoom in, you can zoom out, you can look at the specific annotations, we call them. And annotation, if we go back to the analogy of Google Maps, right? So you have the street map. And then on top of that, you have a layer. For instance, you can see traffic. So that's a layer of annotation on top of the coordinates. And that's how it works. You have the actual reference genome, and then a layer on top could be start and end positions of genes. You can put on top another layer of, for instance, regulatory regions, where they are. You could put another layer on, for instance, specific mutations that have been associated to a particular phenotype, let's say. So we have layers, which in the genome browser will look like tracks. When you look at the I don't know. I can't think right now of an analogy, but basically you have you have like, we call them tracks, and you can configure those tracks to show you just a raw sequence, to show you, well, another track where there are known variants from specific studies and so on. So in other words, this is free again, and this is really useful. Having these data resources enables the infrastructure to then for people to use them as a tool for generating discovery. Someone I know was equating this kind of infrastructures to motorway, right? So if you have a motorway, you can travel relatively easily. So the reference data sets and the reference tools that we have allow us to navigate scientific knowledge, biological knowledge, in a way that enables people to journey through the different types of knowledge that are accessible. Have you guys heard of Moore's law? You know who? You know Intel, right? You know Intel. So Moore's law was, so there's I think his name was Gordon Moore. He was the CEO of Intel, and then in the 60s he suggested that the processing capacity of transistors, which are basically the underlying structure of computational processing, doubles every 18 months. Okay, so it's been a quite predictable pattern that has happened throughout decades, still happening. You know, just compare, you know, iPhone 7 with iPhone 13, right? Or iPhone 8. Now it's iPhone 16, I think it is, right? So compare iPhone 16 to iPhone 15. You know, it kind of doubles every time, every 18 months. So we see that sort of evolution of processing capacity, which is exponential, right? Because, you know, it doubles every 18 months. So if we compare Moore's law, which is this doubling of processing power, with the how the cost of generating a whole human genome has decreased, we can see that approximately a genome today, sequencing a genome is 50,000 times cheaper than what it was 20 years ago when we had the first human genome. All right? So yes, it's very expensive still for most of the world. But the type of exponential explosion, should we say, in terms of how much it costs. Cost obviously is probably the most important factor in making genomes accessible to people, but the rate at which the cost of producing a genome has decreased has been exponential, and is 50,000 times cheaper. So what that means is that if you can produce this kind of very valuable data a lot cheaper, then the production of data coming from genomics has exploded in the past 20 years. So I still remember the first release of the human genome. Then in 2008, there was the 10,000 genomes, UK 10K. Then in 2014, then we had the announcement by David Cameron. You remember David Cameron? The 100,000 genomes. And by the way, it's astonishing that the UK has been at the absolute forefront in the production of and the development of genomics in many different ways. So then in 2018, we had the 200,000 release of exomes from the UK Biobank. Exomes. I talked about exons, and I'm saying exome. So if a gene if all of the genes in a cell we call a genome, what do you think an exome is? All of the exons in a genome, right? So exomes were released for 200,000 people via the UK Biobank, and guess what? Last year they released 500,000 genomes, whole genomes for research, and that has never happened. So you see that in 20 years, the pace at which things advance, they double, they triple, every few years. So I don't know. I can't comprehend how people 2 years ago could live without ChatGPT. Do you use ChatGPT? Come on, raise your hand. Is there anyone who doesn't use ChatGPT? You don't use it? Everyone uses it. You guys haven't said it. You guys use it? ChatGPT? And you too? Of course not. No, for your course. Of course not. I would never assume that. Of course. Isn't it incredible how things are advancing? I've got news for you. It's going to get faster. You know that, right? It's going to get faster and faster and faster and faster and faster. And I'm going to get younger and younger and younger. Hopefully. No, maybe. But anyway, that wasn't a good joke. So genetics, genomics, some people call it synthetic, but biomedical sciences and computer science, right in the middle. Everyone that is saying about the future is kind of the convergence between the silicon and the genetics, into something new, right? So these are genomics has become one of the leading sciences pushing the boundaries of computing. Okay? So instead of calling astronomical datasets, you could call them genomical datasets, because the explosion has been so massive. And this is what it looks like in its digital form, right? So that's actually a bit of my genome, which I've sequenced 5 times already. It's just like screenshots of the raw sequence of my genome, and here it is. This is it. This is real. This is not fake, I promise. This is real sequence of a supposedly human being, certainly a fellow specimen of your species. That's a real bit of someone's genome. The book of life, they call it. The book of life. Except that, at some point is, the analogy and the metaphor of the book of life kind of breaks. A book has chapters. Okay, let's say that a chromosome is a chapter in the book of life. But then, a book has sentences, paragraphs, full stops, commas, punctuation, spaces. There's no punctuation there. And yet, somehow, DNA polymerase is able to start transcribing at the right position within the right time in the right moment. How fascinating is that? How is that machinery inside every one of the trillion cells is able to specifically transcribe within there, somewhere here there's a stop codon. So somewhere here there's a start codon. Somewhere there's an intron. There's an exon. There's a regulatory region. Somehow there, I can't comprehend. They all looks the same to me. And yet, that combination of letters is going to explain why I have diabetes, why my father got prostate cancer, why my aunt got melanoma. Somehow, that combination of letters relates to you having illnesses. Okay? So there's a lot of intermediate layers between that raw data and what you see at the level of the phenotype that we're still trying to comprehend and integrate. We're just at the very beginning of the journey. But somehow, all of that information, at some point you were a zygote, right? We all were a zygote, just one cell, sperm and an egg, just one cell. You don't see, and we look like our parents. We inherit the diseases of our parents. That was there. That information was there. And it was there. So, this is just a summary of the length of nucleotides. Hopefully you all know what a nucleotide is, right? Do you know what a nucleotide is? Everyone needs to know what a nucleotide is, otherwise I'll define what a nucleotide is. A nucleotide is a chemical compound, adenine, thymine, cytosine, guanine, that composes and contains that forms basically the DNA chain. So you can see all of the chromosomes. Can you guys see this at the back? So you can see on the left-hand side in red the number of genes. So you can see that in general, the longer the chromosome, you can see that chromosome 1 is in green. Chromosome 1 is longer. It has 250 million base pairs. Base pairs is basically you know this, right? The DNA has two chains. So base pair is the position of the two chains. Let's say CT or GC. A always binds T, G always binds C, right? So base pairs, we call it here, because we are talking in the context of double helix, double chain. So you can see that chromosome 1 has 250 million base pairs, and it goes down to chromosome. Oops, they got it wrong. Because they thought at the beginning that chromosome 22 would be the smallest, but actually now we know that actually the smallest one is chromosome 21. So they got it wrong there in the way they named chromosomes, but anyway, there's nothing completely absolute in biology, nevertheless. So what you can see is that actually, although the number of genes per chromosome tends to diminish with the size of the chromosome, but there are some genes that are more gene rich, and some chromosomes that are more gene rich, and there are some chromosomes that are more gene poor. Look at, for example, chromosome 18 has fewer genes than the smallest chromosome, which is chromosome Y, by the way. We've seen this. I'm just going to reinforce what I said earlier. So we have the genetic code now from a person in its digital form. I also find this miraculous. That's basically somehow you put a sample, saliva sample, DNA extracted sample into a machine, and then suddenly it becomes digital. So you put something material and then it converts it into some kind a file. Yeah, so there's magic happening there. File digital information which then we can analyze, we can process, we can quantify. So I said earlier that a variant is basically a change in any of the sequence that we get in a patient or an individual or an organism versus its reference. So if at that position in the reference, instead of being a T, is a C, and here we have a T, then that's a variant. Okay? So I want to stress that I'm not talking about mutations, right? Mutations is the world of genetics, right? We're now in the world of genomics, yeah? Digital, big data, processing, computing power, Moore's law, yeah? That's our world, my world, your world now. So, and we don't talk about mutations anymore. We talk, that's for Mendel's laws, which were fantastic before the digital age. And in the digital age, we call variants. We don't call mutations. So a variant is a change between the reference, consensus reference, and what you observed at the level of the individual. So you now know what a SNP is. So here is an example of a SNP, a single nucleotide polymorphism. So it has a beautiful name, RS429358. As you can see, it's very memorable. So that's actually is my favorite SNP of the it's my second favorite. It's my second favorite SNP. It's not my top favorite SNP. And I'm not going to tell you what my favorite SNP is. You have to see me in a different lecture for the future. So I'm leaving that there. Yeah, suspense. For next time you ever see me, just ask me about my favorite SNP, which is, by the way, my favorite gene, melanocortin 1 receptor. I'll give you that. So, but you've heard of a gene. I bet you have heard of this gene, ApoE. Have you heard of that gene? ApoE. Have you heard of that gene? What's that gene? Where have you heard of that gene? What do you think it does? Okay.
I know it's related to Alzheimer's.
Oh, right. Okay. Okay. Right, very good. So we have a gene called ApoE that apparently is associated to Alzheimer's. All right? Okay. So my second favorite SNP in the whole world, RS429358, happens to be located in chromosome. Can you guys see this? It doesn't look very nice. Can you kind of read it? So I've got this SNP, my second favorite SNP, that is in chromosome 19, position, I'm going to breathe, position 44,908,684. Right, so at that specific position within the 3.2 billion letters, at that specific position, there is a SNP which consists of a T being changed for a C in that position. All right, so I'm introducing you to your first ever date with a SNP. This is your SNP. Yeah? So, and it happens that is inside an Alzheimer's gene, probably the most important gene in Alzheimer's. Right? And it says it has a consequence. So you make that change, it has a consequence. And what consequence it is a missense variant. What's a missense? Sorry?
Would it be a stop?
That would be a stop codon. A missense, but it's on what a missense does. What's a missense mutation? Changes the amino acid. Right? So we'll get back to this second favorite SNP of mine. Now, we are lucky because that SNP, we know what it does. Somebody has painstakingly performed analysis, a set of validation experiments and so on, and is able to tell us what the consequence is and what it means, right? Interpreted. But whenever we see this kind of variant, any variant, these are the two questions that we are interested in. One, does this sequence change in the correct genetic background provide an explanation for phenotype in an individual, right? This is what I said before. Is this genotype producing a particular phenotype? Is this particular variation associated to a specific trait that I observe? Yes or no? And then the second question I'm interested in, is an individual who inherits this sequence change likely to develop the same trait condition phenotype? Right? There's something called the American College of Medical Genetics and Genomics, abbreviated as ACMG, that tells us that we can classify the effect of a given variant. And it gives us five choices: pathogenic, likely pathogenic, uncertain, significant, likely benign, and benign. So it's kind of a grade, it's like fail, third, 2, 1, and first, something like that. So, which is rather, I don't know, for some people inconvenient and simplistic, but here we are. This is what we have. So we have a set of standards for classifying the effect that a particular variant has in the individual, and it goes from more severe to less severe. So pathogenic contributes to the development of disease, although some changes may not necessarily be fully penetrant. Have you come across that term? Penetrance. Somebody is nodding here. So can you define for me what penetrance is? With your own words.
I think it's if the sequence actually leads to a physical presentation or the expression of the condition.
Yes, so exactly. So it's whether, so you may have a fully penetrant change, means that everyone who has that change has that trait or that disease or that phenotype. But that never happens. Okay, some of the most penetrant variants that we have are in BRCA1 and BRCA2 genes. You've heard of those, right? and the penetrance is around 80%, meaning that 80% of carriers of that mutation express the disease. So even the most fully penetrant are not 100%, especially for polygenic diseases such as breast cancer. Any questions? Is it clear so far? So we've seen now, we now know what an exome is, right? Can someone remind me what an exome is?
All of the exons.
Yeah. Which happen to be around 2% of the whole genome. So only 2% of the whole genome is actually coding. Can you imagine? Yeah? So what does the rest of the genome do? What do you think? There are regulatory regions. What else? Structural functions. But guess what? We also have viruses inserted there. We have repetitive regions. We have lots of what people used to say junk, which actually is not junk, because according to ENCODE, which was one of the big projects trying to assign function to every bit of the genome, they affirmed controversially that 80% of all bases of all positions in the genome can have a functional annotation to that, to them. That doesn't mean that they have a function. It means that potentially we can assign specific features, characteristics that could be associated to function to about 80% of the whole length of the genome. And there are gene deserts and gene-rich regions. Can someone tell me or guess what bit of the chromosome would be a gene desert?
Telomeres?
Yes. Telomeres, centromeres, repetitive regions. So the 2% of a whole genome occupies just the raw data. I mean, not all of the data, but just the... Actually, so if you take the data that comes out of the sequencer, you know what a sequencer is, right? So these machines that do the magic of transforming the DNA into some kind of digital file, right? So the sequencer produces raw data files for an exome, which I said is 2% of the whole genome. It occupies around 4 gigs of data. Yeah. You had a question?
Yeah.
And the whole genome, raw data is 200 gigs. Try downloading that. Yeah? And if you have 1,000 genomes, how much that's that? 200,000 gigs, which is 200 teras. 10,000 genomes. No, hang on. No, sorry. 200,000 gigs. Yes, it's 200 teras. So that's 1,000 genomes. 10,000 genomes. That's 2 petabytes, right? Is it? 10,000 genomes is 2 petabytes, right? 100,000 genomes. That's 20 petabytes. 500,000 genomes. How much did we say is 100,000 genomes? 100,000 genomes is 20 petabytes. Okay, 100 petabytes. Right. So the UK Biobank, just on genetic data, contains more than 100 petabytes. Which is not bad, is it? Considering that your average laptop, how many gigs you have of storage? Your laptop, how many gigs of storage you have? Yeah? 1 tera maximum, I guess.
I think maybe 6.
6 what?
6 gigabytes.
No.
I'm not sure.
You're not sure. It's not 6 gigs, I can tell you that.
100.
Anyway, I think, my big computer at home, how many do you have there? 2 terabytes.
Yeah.
What machine do you have? How much you pay for that? Don't tell me. Must be good, man. You are loaded to pay that. Anyways, can you see the paradigm, what we're dealing with now? And guess what? You're going to be, when you think about this lecture ever again, you're gonna get tired of listening. Guess what? It's gonna accelerate. Exponentially. You know why? Because it's not just going to be your genome. You're going to have your genome, your transcriptome, your proteome, your epigenome, your exposome, your interactome, your wearables. Your wearables. It's going to be everywhere. Wearables. You're going to be, in the next... You already are for Google, for instance. Wherever you are, there's this cloud of data following you, regardless, linked through your Gmail account that you give everywhere. Yeah? And all those cookies you've ever accepted in your life, you are traceable forever. So all of that data will follow you everywhere. And guess what? It has tremendous implications for understanding your health and disease. Okay? Imaging data. Blood tests. Physical measurements. All of that defines how much are you going to age, how likely you are to have prostate or breast cancer, how likely you are to, have a stroke. It's coming. It's here. And all of that scans back to that map, physical map of the human genome, which for the first time we can begin to have a physical stratum where you can map functions that are then passed from generation to generation to generation to generation. I'm talking too much. I've said this more or less. So we know what a genome is, what an exome is, right? So what's the difference? So an exome is cheaper, and it captures 80% of all the functional annotations that we know, which are in genes. But there's a lot of function that happens outside of genes as well. I said that an exome occupies around 2 gigabytes. This is just a very rough estimate. And a whole genome occupies 200 gigs. It costs $1,000. An exome maybe costs around $400. So if in this massive project that I work on called the Deciphering Developmental Diseases, that was the sort of flagship project for understanding genetic diseases in people with rare genomic disorders, we found that the diagnostic yield, by doing just exome, the diagnostic yield of patients with rare genomic disorders was only 30%. So most of the people go undiagnosed. You've seen them, right? You've seen children, they are born, you know this, they're congenically born with something that doesn't look healthy. I hate to say normal. Healthy. Or there might be a problem, right? They are born with it. So and people may say, and this is wrong actually, the genome, whole genome, the diagnostic yield is about 50%, not 70. So pretty much most people who have a rare genomic disorder, even by looking at the whole genome, around 50% in the best case, go undiagnosed. The question I was always asked, what's the point of diagnosis? There's no treatment. There's nothing you can do. You're born with that. Correct. There's nothing you can do. I mean, no, not correct. We can't change your DNA, at least currently, and I don't think it's going to be, no matter what they say, I see quite unlikely that gene therapy is going to work in the near future, unless we have, I don't know. Now with AI, I can never say that. But you know, at least in the near future, we're not going to be able to change our genomes. And so, what's the point of diagnosing if you cannot really do much? Well, first of all, it's halfway towards curing. Having a label, particular disease, being able to say you have neurofibromatosis, or you have Charcot-Marie-Tooth disease, or you have X. That's such a relief for the family, because now you can, the NHS is going to be able to give you, even if they can't cure you, at least you can join parents associations, you can ameliorate symptoms. You know, now rather than being completely blind, trying to hit anywhere and everywhere. Now you have something, what to expect, how to improve the quality of the patient. Just by being able to diagnose, by being able to assign to the patient's phenotypes the specific genetic cause that made that disease. All right? So, very, very important. And even if 50% go undiagnosed, before we had genome or exomes, the diagnostic rate was 11%. So in fact, in the past 20 years, we pretty much quadrupled the number of patients that go and get a diagnosis. Yeah, so now it looks different, doesn't it? It looks different now. In 20 years, we've improved our diagnostic yields 4 times because of that. And most people don't benefit right now when you get your genome sequence. But the 6% of people with disabilities, with rare genomic disorders, who have that, 6%. How much is 6% of 70 million? Yeah, couple of million, 3 million people, right? That are directly benefiting from this. Yeah, it makes a difference. It makes a real difference to a lot of people. We know now what genotype versus phenotype is. You guys know what dominant and recessive is? Can someone refresh my very fragile short-term memory, please? What's a dominant gene?
Dominant is something that will always show up as long as you have one copy.
Okay.
And recessive, you need 2 copies, and it can skip generations and hide.
Cool. Thank you. So, you guys have looked at complex diseases, polygenic diseases, in any lecture before? No? Okay, cool. I understand you've looked at monogenic diseases, right? So diseases that are caused by one mutation. You looked at that? That's what we call Mendelian disease. So here we have Mendelian disease and complex disease. So Mendelian disease are caused by a mutation in one gene. They are rare, they're highly penetrant. They can be autosomal dominant, recessive, so they have Mendelian inheritance. And they're minimally affected by environmental influences. The vast majority of what people die of is polygenic diseases. Yeah, I told you these 6 diseases. Type 2 diabetes, atrial fibrillation, coronary artery disease, inflammatory bowel disease, prostate cancer, breast cancer. 25% of people die because of those. And they're highly, highly polygenic. That means the likelihood for you to develop the disease has a significant contribution from the variations, so from your genetic makeup in many different genes. Okay? That's what we mean by polygenic or complex diseases. And there are common diseases. The penetrance is variable, very variable. There are no clear modes of inheritance, although I've been looking at how when you have, let's say you have a mother with high polygenic score of diabetes and a father with low polygenic score of diabetes, what I find is that they tend to compensate. So they don't follow the normal pattern you would expect in Mendelian diseases. And environmental factors have a tremendous contribution. So they are much more affected by the environment than rare diseases, because you are born with those, right? So here we have an example of two complex diseases. So type 1 diabetes, the genetic contribution is very high. That means that pretty much it doesn't matter what you eat. You know, if you have high susceptibility for type 1 diabetes, most likely you're going to get it. The opposite end of the spectrum is melanoma, where you have not so much contribution to have melanoma is a type of skin cancer. So basically, if you have high genetic predisposition for melanoma, don't go to the beach, right? Or have a lot of protection, or both. So there's something called genome-wide association studies. Have you heard of that before? Genome-wide association studies. So genome-wide association studies, what they do, they work with SNPs. Yeah, we now know what a SNP is, don't we? So the way they work is basically a statistical model, whereby in this hypothetical case, so we have, let's say, cases of breast cancer, of women with breast cancer from the same genetic ancestry, from the similar location, all things equal, with women of the same type, but they are healthy. So controls, we call them. If we find SNPs, as symbolized here by the red marker here, we're going to find that, of course, the same red SNP is present in both, but actually, if the frequency of the red SNP is significantly different from the frequency of the red SNP in controls, cases versus controls. So here we have cases, they have 60% have the red SNP, and controls only 16% have this red SNP. So statistically speaking, there are the frequency of the red SNP, actually allele, it's not a SNP, it's allele, because the SNP is the same. Sorry, it's the red allele. Sorry, I should say red allele. So the red allele is significantly more frequent in cases than in controls. So statistically speaking, then if we see the red allele in a person, then we say that they are more likely to have susceptibility to breast cancer, let's say. Is that clear? So that's how it works. Except that for polygenic diseases like breast cancer, we look at all millions of SNPs. We don't just measure one, we measure millions, and then we compare the frequency for each SNP between cases and controls. And we end up, from those millions, maybe we end up with 6,000 that are significantly associated to presence of breast cancer. Yeah, so that's really what it is. And we call this genome-wide association studies, GWAS for short. And not just that, we know that different SNPs have different weights. So some SNPs, some alleles are going to contribute more to disease or to risk than others. So we can assign them weights. And which is what we call risk scores. So if we are able, for the, let's say, the 6,000 SNPs, you're not going to have all of the alleles, all of the risk alleles. You know, you're going to have a distribution in the population by summing all the weights of all of the increased risk alleles. You know, if you could imagine each blob is an individual, right? And you are basically summing the weights of observed risk alleles in each individual, you're going to end up having a normal distribution. Do you follow this? So basically what we're doing, so each individual, you sum all of the weights of observed risk alleles. And now you plot them in terms of how much risk they have. So you're always going to have a normal distribution, which means on average, most people are going to have average risk, right? So this is what it means. And few people are going to have low risk, and few people are going to have high risk. So by summing the contribution of each individual observed risk allele, we can actually stratify high-risk individuals. Yeah? I use this very technical specific term, stratification of high-risk individuals. And that's how genomics can be used for complex diseases, to stratify high-risk individuals. You remember my second favorite SNP earlier? RS42938. So if you have that tiny little change of a T into a C, it increases your risk of developing Alzheimer's 253%. Yeah, there are 3.2 billion letters. One little tiny change. Now, the vast majority of the SNPs do nothing, okay? We all have hundreds of variations, millions, but a very few can have a huge impact, okay? So, wrapping up. Unless you see me again, this is probably going to be the most data-driven type of lecture you're going to have for a while. And I can not emphasize enough that medicine and biology are technology and data-driven today. Yeah? I can't emphasize enough that you need to be computer literate. You know that. I know. But I don't mean using. I don't mean using apps. Yeah? I don't mean using the apps. I mean potentially developing even. If you have data skills, you're going to have a tremendous advantage. I would even say a job. More chances of a job than your other fellow biologists, biomedical scientists. Right? I know I'm biased, but I can tell you that it's made a difference. It's just a sample of one, but I can tell you, I could even beg you to think about this. I'm going to say something, my second controversial statement of the day. Most of the biology you guys are learning is obsolete. If you don't put it into the context of, I'm going to nuance it, if you don't put it into the context of the fact that now the bottleneck is not the production of data. The bottleneck is the interpretation of the data. the analysis of the data. Yeah? Is go beyond Excel. Yeah? Big data, my definition of big data is whatever whatever analysis you want to do that an Excel spreadsheet cannot hold, right? Maybe, let's say 100,000 rows. Yeah? You have 100,000 rows on a table that you need to analyze, then we're talking business here. I genuinely say, because I truly believe it, that that's going to make a difference in your chances of being employable. And I don't believe the crap that they say that AI is going to take all our jobs. No. It's going to take the jobs of those who don't harness AI. Don't fight the wave, ride the wave. Surf the wave. Yeah? Don't fight it. So I believe that genomics is going to be an indispensable tool for medicine. Now, I've talked about genetics and genomics. This is not a, genetics and genomics is not a crystal ball, magic crystal ball that is going to tell you the future. It isn't. It can only give you a probability of something. And it's only one of many contributions that you can have to understanding of disease. But it's a new addition. It's an independent data point together with family history, with your measurements, your wearables, your analysis, blood tests, whatever. But it's a very valid new type of information that certainly has a contribution. All right? And my bet is that everyone is going to have their genome sequenced in the next 10 years in the NHS. And you haven't heard about this, but they're starting already with a newborn genome sequencing. They started already doing 100,000 newborns. So all newborns are going to be genome sequenced very, very soon in this country. By default. Okay? And you have the opportunity to ride that wave as well. Okay? And don't expect things to be simple. They're going to become even more complicated. Yeah? Biology is complex. We've only started to scratch the surface of what's coming. We have no effing idea of how life works. We don't understand biology. We don't understand the mechanistic processes that influence health. We're just beginning to understand a little bit. This is the work of a lifetime. All right? So anyway, I'll leave it there. Thank you for your attention.